Welcome to our video on sedimentation velocity of interacting macromolecules. The material stems from our workshop Biophysical Tools for Studying Protein Interactions, which we held annually at the National Institutes of Health and hosted by various research institutions. But we wanted to make this lecture more widely available and spread the word of how sedimentation velocity works and how to apply it, because it's really a very powerful tool to study protein interactions in solution. I will discuss a mixture of basic concept, theory, and how that determines the practical applications. We will go through a number of topics. After a brief recap of the experimental setup, we will talk about the master equation of sedimentation velocity, and then inspect the different types of boundary patterns that we encounter in sedimentation processes of interacting systems. This will lead us to dive into some tools for quantitative data analysis and for how to understand what's going on. And this will bring us then to a practical framework for effectively generating and analyzing experimental binding isotherms. We will then discuss an independent approach for studying multi-component macromolecular complexes through a multi-signal extension of our sedimentation coefficient distribution analysis. And finally, we'll talk about how hydrodynamic interactions impact the sedimentation process and how we can take that into account. This is a long presentation, but the advantage of a video instead of a live lecture is, of course, you can pause it at any time. For example, since the material builds on foundations described previously in this series, we will show references to other videos that you can consult if you wish before continuing. Up front, if you're unfamiliar with sedimentation velocity, I would recommend to start with these two videos. Likewise, you may pause the video to carry out some practical exercises using data and software provided through the workshop. Having more time than in a regular lecture gives us also a chance to add a little bit of historical perspective, which in my view can add a lot because this topic has been studied for almost a century. And I apologize upfront for any omissions in this regard, which will be many. I will occasionally provide some references to the literature, but mostly our own papers, from which you can find more context and technical details. So, as with most of what we talk about in this workshop, you don't have to worry about solving any equations. This is embedded in our software ZFIT and ZFAT, but it is essential to understand the concepts. As a companion material, I would recommend our book on interacting systems, which contains a rigorous theoretical backbone along with detailed explanations of the software functions that help carry out the difference analysis. You can also see some of the figures examples in the video are actually taken from the book. Finally, there are user groups which you can join to post questions or discuss issues related to the biophysical techniques, the software, or any specific application. So let's start by recapitulating the experimental setup of sedimentation velocity. In the experiment, we have a sample holder in the spinning rotor and optically measure the concentration distribution as it changes with time as a result of the applied centrifugal force disturbing the sample. And for interacting molecules, we almost always try to carry out the experiment such that there is chemical equilibrium at the start of the run. As we have seen before, this typically produces sedimentation boundaries that reflect migration from sedimentation and diffusion. And I like to visualize this as brown in motion with a bias. So you see these little dots whizzing around with a bias. Now, for interacting systems, let's say monomers are blue dots and the faster sedimenting complexes are the red dots. Now, if the complexes are not stable, they may dissociate. But they can readily reform since they always remain in an undiluted bath of their slower sedimenting components. So how can we use that for studying protein interactions? So perhaps the most straightforward approach from a theoretical perspective would be to use Lamm equations to describe the sedimentation process of the individual species and their complexes that are formed. In fact, at least that was the big hope during most of the 20th century, when solving the Lamm equation numerically or with analytical approximations was not yet feasible efficiently enough for practical data analysis. And so this approach remained theoretical and still seemed very appealing. You may have seen before in this lecture series how accounting for sedimentation and diffusion fluxes in the sector-shaped cell leads to this master equation of sedimentation velocity. It simply describes the sedimentation patterns of different size molecules. And I have already mentioned that once we were able to easily compute Lamm equation solutions in the 1990s, they turned out not all too useful for directly analyzing most real-world samples with their preparative imperfections or sometimes pleomorphic and polydispersed nature. The need to avoid fitting discrete Lamm equation solutions led us to develop the sedimentation coefficient distribution C of S, which is now very popular. Nonetheless, it is worth looking at extensions of the Lamm equation for interacting systems. There are basically two approaches. The first is particularly useful for self-associating systems, where the population of different oligomeric species is completely determined by the total concentration of the macromolecular component. Assuming that chemical kinetics is very fast compared to sedimentation, then locally in each little volume element there will be chemical equilibrium achieved quasi-instantaneous for the purpose of sedimentation. Now, because conveniently we measure the total concentration as a function of space and time, this allows us to calculate the local populations of all species at any point in time. And so we can make use of the fact that sedimentation of the system is governed by the local weight average of all species and diffusion by the local gradient average of all species. And then we can fit this to the experimental data. And the result of such an analysis are the S values of all species as well as the equilibrium constants based on mass action law. Now, the second approach is useful for heterogeneous interactions and/or for systems where reaction kinetics is comparable to the timescale of sedimentation. Here, instead of taking local averages at all radius positions, we keep track explicitly of all species and how they chemically interconvert. And this produces a system of Lamm equations that can track each species' sedimentation and diffusion fluxes as well as the chemical reaction fluxes. These tools are implemented in ZFAT, and here is an example of how this can work. In a collaboration with Roy Mayusa's lab, we studied in this project two peptides, one of which has significant absorbance at 280 nanometers and the other one doesn't. So, by acquiring data with absorbance system and the refractive index sensitive interference detection system, we can get additional information about the concentration distributions of each component. Never mind that there's also some unrelated signal offset from sedimenting buffer salts in the interference data. Then we fit the Lamm equation solution accounting for the two free protein species and a one-to-one complex. You can see the red lines from the best fit model with an explicit Lamm equation fit matches the data points really very nicely. In this case, the best fit equilibrium constant is 10 micromolar and the complex is quite long-lived with an off-rate constant of 10 to the minus 5 per second. We will come back to this example later in a different context. Here is another example, also from a collaboration with Julie Lamm and Roy Mayusa's lab. The interacting proteins are a natural killer cell receptor LY49C binding MHC class I molecules. The crystallographic structure showed two molecules of MHC binding to one LY49C, and so the point of the experiment was to see if the interaction really exists in solution and if it also has a 2-to-1 stoichiometry. The data were collected for 10 different concentrations with LY49C held constant and MHC concentration titrated in varying concentrations. The data for 3 of the experiments are shown over here. It is worth noting the boundary structures. They show sedimentation boundaries at equal time intervals so that we can better compare visually the relative velocities. On top, with an excess of LY49C and just a little bit of MHC, we can recognize two boundaries, both relatively slow. When we have just a little bit more MHC than LY49C, then we see much faster sedimentation, in what looks to me also a bimodal pattern, with maybe a little bit of a boundary in the trailing part here that might be similar in velocity as the slow boundary here. In any case, at maximum molar excess of MHC, we see no slow boundary at all and just two very fast boundaries, one sedimenting ahead very quickly and then another one over here which might be similar in speed to that one. But if you think about it, this is really quite strange. There are 4 different species in solutions. Free Ly49C, free MHC, maybe a 1-to-1 complex and probably a 2-to-1 complex. But in any case, here we see only 2 sedimentation boundaries at any given condition. Without thinking about this further, we can go ahead and throw a Lamm equation model at this. The fit is shown again as the red lines, and the fitted parameters are shown here. You can see that the model fits the data quite well. The KD was 1.7 micromolar, which is close to what we obtained from sedimentation equilibrium. And the sedimentation coefficients make sense based on hydrodynamic modeling with HydroPro. The RMSD is very nice, 12 millifringes. Now the K-off was pretty fast, 0.1 per second at the best fit estimate. Now we can explore the same data with different models. If we use, for example, a single-site model, then the fit gets much worse, almost twice the RMSD, which means that with the Lamm equation modeling as a tool, we can discriminate between the right and imposter models. However, if we vary the kinetic rate constant and impose a slower K-off to this model, then the fit is almost as good as with our best fit K-off. Only K-offs that are drastically slower by 4 orders of magnitudes here produce a very different fit. So if you happen to watch this video as part of the workshop and want to practice hands-on analysis, at this point you can pause the video and find these example data sets and carry out some exercises. We will try to make these data available with tutorial material also on our website so that everyone can follow along carrying out the analysis on your own computer. Let's summarize the pluses and minuses of direct boundary modeling. Some of the advantages are the theoretical appeal, the fact we can incorporate diffusion information in the model, that it lends itself to global modeling of data at different concentrations or even acquired at different optical systems, and that generally we can, in principle, incorporate any type of prior knowledge into the model, and that it will allow us to extract estimates for kinetic parameters and equilibrium constants as well as S-values of all the species. Now, on the downside, these kinetic parameters are often poorly determined by the experimental data. And if we have a very complex system, the model can be difficult to build and verify and is often very time-consuming to fit. I want to stress a key point. In our experience, fitting a single discrete Lamm equation to data from one non-interacting protein usually does not work well due to the exquisite sensitivity of sedimentation velocity to microheterogeneity and even trace impurities. The same problem persists here in the analysis of interacting systems because still the models ascribe one discrete Lamm equation solution to each species. They are just now coupled by reaction terms. In fact, the problem is compounded here because now we require homogeneity of multiple protein preparations at the same time. And any imperfections will lead to a bad fit. And that leaves us hand-waving with the interpretation of what it means. If we have a model that doesn't quite fit the data, does it still reflect the major aspects of the interaction or not? But for me, the biggest problem is that Lamm equations are basically computational recipes for the evolution of concentration profiles with time. And as such, they describe, but they do not explain what is going on and why we have certain boundary features. As we will see, sedimentation boundaries of interacting systems are very feature-rich and have patterns that are clearly visible to the eye. And we want to understand their physical origin and utilize for the analysis robust models that rest on the strength of sedimentation velocity, which is the separation of mixtures. And we want to use models that allow us to exclude contributions from unrelated sample imperfections should they occur. Okay, so in the next section I'd like to go through zoology of sedimentation boundary patterns of interacting systems. That will help us to develop some intuition, and in some cases it'll allow us to draw qualitative conclusions just from looking at the data. And it will also set us up to develop some quantitative physics-based models for the sedimentation process. And if you haven't been working with CFS analysis so far, then this would be a good time to catch up with this, because we'll be using CFS analysis a lot in the following. Up front, I think we all agree that the sedimentation coefficient distributions CFS provide us with a great representation of the sedimentation boundary patterns. Because they deconvolute diffusion, they home in on the boundary velocities and amplitudes, and this is the most robust information content of the experiment. As we have discussed in the context of non-interacting particles, interpretation of the diffusional spread is susceptible to microheterogeneity and preparative imperfections, and that's why we want to disregard that for now. So here, the fact that there are two sedimenting species is quite obvious when the velocities are very different. But in this case, they are more similar in their velocity, then the diffusional spread really covers up their differences in velocity, and it's hard to recognize that there are two species. On the other hand, the CFS analysis can deconvolute diffusion and show us the S values of the two underlying species nicely. Now, let's look at the boundary patterns as a function of lifetime of the complex, or in other words, the kinetic rate constant. To examine this, we have simulated sedimentation profiles using Lamm equation solutions for either a heterogeneous bimolecular reaction A plus B forms AB, or for homodimerization. We picked parameters for some moderately sized proteins at concentrations equal to the equilibrium dissociation constant, so that we have both free and complex species nicely populated. And then we plugged in different kinetic rate constants. Now, we would typically sediment such a system at 50,000 rpm in a 12 mm solution column to generate nice boundaries. And that's what we simulated. We then carried out a CFS analysis on each of these datasets for all rate constants. On the bottom here are shown some representative CFS profiles, left for the heterogeneous reaction, in here for the self-association. Obviously, it can get quite busy looking at such superpositions of many CFS curves. Therefore, we have turned them into heat maps, where the S values are plotted from left to right, and the height of the CFS distribution is turned into a color temperature. And in the vertical direction now we can plot the different kinetic rate constants, which we changed in the underlying simulations for each CFS trace. And so, for very slowly dissociating systems, here with rate constants below 10 to the minus 5 per second, the lifetime of the complexes is on the order of days, whereas the centrifuge experiments is just a few hours. And so, in relation to the sedimentation process, these complexes are essentially stable particles. They just sedimented their characteristic S values, and we can resolve them. So you can see here that the species A and B and the complex AB are nicely hydrodynamically separated. And similarly, for the dimerization system, we can separate the monomer from the dimer. But as we turn on the dissociation rate, a different behavior starts to show when the complex lifetime is less than an hour. Let's look at the dimerization first. For any rate constants larger than 10 to the minus 3 per second, corresponding to a dimer lifetime of about 10 minutes or less, we see just a single boundary, and the S value of this boundary is between that of the monomer and the dimer. Now, it's easy enough to imagine what's happening. On the time scale of sedimentation, all protein copies rapidly exchange their state between monomer and dimer in a rapid association-dissociation equilibrium. While they are monomeric, they sediment slowly, but when they are in a complex, they sediment fast. And what we see macroscopically for the ensemble is just their time average S value. And that's this. Now, things get a little bit more interesting for the A plus B system. When they approach the rapid association-dissociation mode, then they will show two boundaries, one at the S value of A, and the other at an S value in between that of B and the complex AB. We will spend some time later discussing what exactly is going on. But for now, I just want to point out that there are basically two types of patterns, those of slow interactions and those of fast interactions. Furthermore, we can recognize that the transition between these two sedimentation modes happens over a quite narrow range of kinetic rate constants, when the complex lifetimes are just about 10 minutes to half an hour, or k of between 10 to the minus 3 and 10 to the minus 4 per second. Now, this transition would change a little bit if we were to use very different roller speeds. We can already see that there is little hope to determine off-rate constants from our sedimentation experiment outside this narrow range. And here we are looking at the concentration dependence of the boundary pattern for the same systems. The C of S distributions are here normalized and then again color-coded, and now we are plotting in the vertical direction the log of concentration relative to Kd, ranging from 2 orders below Kd to 100-fold Kd. In the lower row is the case for slowly equilibrating systems. Clearly, we can hydrodynamically separate the different free and complex species in solution and observe their shift in relative population as predicted by mass action law. Now, for rapidly equilibrating systems, on the other hand, we see again some gradual shift in S values. For homodimerization, this starts at the monomer S value when we are far below Kd. As we raise concentration, the boundary S value increases because the fractional time each molecule spends in the dimer state increases. And this goes until we virtually saturate the assembled state. Now, in the case of the heterodimer, I should mention that the simulations assume equimolar concentrations of A and B. Far below Kd, there is very little complex, and we see free A and free B, but as we populate more complex, the faster boundary increases in velocity, while the slower boundary stays put. But it fades out in comparison to the complex as we increase the saturation of the system. So you have just seen that for rapid monomer dimer systems, there is just a single boundary with the velocity dependent on concentration. But things can be a little bit more complicated for higher self-associating systems. Here you can see sedimentation boundaries of monomer trimer and monomer tetramer systems. In the upper row, concentrations far below Kd, and in the second row, far above Kd. Below Kd, things are pretty simple because there's not a lot of oligomer formed, and molecules remain mostly monomeric and sediment close to the S value of the monomer. At higher loading concentrations, however, you can see that the boundary can split. This is because in the diffusion-broadened boundary, the molecules at the trailing edge will see a lower concentration and therefore dissociate from the oligomer into monomers, whereas the rest of the system sediments at a time average S value corresponding to the equilibrium population at the loading conditions. This is usually not so pronounced for monomer dimer systems. Here are corresponding C of S profiles for concentrations, including these and intermediates. But for monomer trimer and monomer tetramer system, this is very clear. The boundaries have substructure. Okay, we know enough at this point to be able to design experiments and carry out basic diagnostics. First, we should always run samples at a range of concentrations. Then we determine C of S distributions and overlay them. For example, overlays may be done conveniently in the graphing software Gussie from Chad Brodigan. This is a great companion program to ZFIT and ZFAT for graphics and several utility functions. From looking at the overlay, you can then decide whether the peaks remain at constant positions independent of concentration, in which case it is a slowly equilibrating system, or if the peak S values move with concentration, at least some of them, in which case we have rapidly equilibrating systems with fast kinetics on the timescale of sedimentation. This choice will then lead to different analysis strategies downstream, as we will get into. Let us focus a bit on the rapid kinetics case, since this was so mysterious. So I want to show you some more examples of counterintuitive behavior. Let us consider, for example, a mixture where bispecific antibody A can bind ligands B and C. So in this case, we have a total of 6 species in solution: free A, free B, free C, AB complex, AC complex, and a triple complex ABC. And we pick equilibrium constants so that these all are populated. When we solve the Lamm equation for this system, whatever concentrations we pick for the mixtures, we find that there are never more than 3 boundaries, sometimes only 1 or 2, even though we have 6 species. Let's look at the concentration-dependent behavior of the system, again using C of S distributions as a function of concentration. Our starting point are these particular concentrations, where we have initially a good amount of C and just a little bit of A. And then we are going to titrate in more and more B. So we start out with very little B. Initially, there's no boundary of free A, and a lot of it must probably be in the complex AC. On the other hand, we do not have a boundary at AC, but we do have a sedimentation boundary that sediments between the S value of AB and AC. As we titrate in B, we see the boundary gets faster, and I would guess this is simply because some triple complex is forming. And then we gradually build up a boundary sedimenting at the S value of free B. Since we are titrating in B, this all seems to make more or less sense. Now let's look at a different titration, where we start with some moderate amount of B, again a lot of C, and then we titrate in more and more of A. Let me just show you what happens. If we increase A, at first there is not a lot of change, but we notice that increasing A causes the S value of this boundary to decrease. And that is weird, because having more A will certainly allow more complex to form. After all, this is the binding partner for both B and C. Yet the system here sediments slower. This keeps going until this point here where the boundary seems to split. And the boundary for free C seems to get very small, and our fastest boundary changes course as we add more A, and it increases now with concentration. So what is going on? How come that increasing a concentration of a binding partner can cause the sedimentation boundary velocity to decrease, and in the course of this titration then to suddenly change course? Observation of these mysterious sedimentation properties of reacting systems have a long history. They are described in Fujita's monographs and Gilbert's seminal papers from the 1950s. They recognize that two-component systems have no more than two boundaries, even though there are three or more species. The slower boundary is concentration independent at the same S value as one of the free components, and on the other hand it shows a discontinuous transition to a different free component with a transition point that is not at the equimolar point. As far as the fast boundary is concerned, the S value was recognized to be concentration dependent, and that increasing concentrations may decrease its velocity. Finally, at certain concentrations there is only a single boundary at an S value not corresponding to any of the species in solution. This has completely baffled scientists working in this field. It was confirmed theoretically using numerical Lamm equation solutions or Gilbert-Jenkins theory in the 1950s. However, since Lamm equations describe but don't explain, the mystery remained and led to complete rejection of the possibility to interpret reaction boundaries in any way. We have more recently added to this list of strange properties, showing that multi-signal analysis of the reaction boundary shows a composition that may not be that of the complex, and that things even get weirder if we allow for non-additive signals, for example, changes in quantum yield of fluorescence in complex formation or in the presence of hyperchromicity. So clearly we need to develop tools to explain this and hopefully even take advantage of this. With this in mind, the next section is about an interpretation of sedimentation data that is not based on Lamm equation solutions, and it goes back to a time before they could be efficiently solved. Howard Schachmann gave a very nice description of this in his seminal 1959 book, Ultracentrifugation in Biochemistry. Let's say we have a concentration profile at time 1, and then we let the boundary move on to a time 2. And now we ask, what is the mass balance? Which means how much material has crossed some hypothetical line in the solution plateau. Well, that's easy to answer by just integrating under curve 1 and curve 2 and taking the difference. We can be sure that this is exactly the amount of material that must have crossed this line. If we divide this by the time difference, then it'll represent a flux, and then dividing by the total concentration, we can use this to define a weighted average S value. It is important to realize that the molecules we are ascribing this S value to are really the ones in the plateau, since these are the ones crossing the line. And therefore, it's completely irrelevant what the boundary shapes are. Another consequence is that since we are only integrating the concentration profiles, any model that faithfully describes the boundaries will lead to the correct integrals and the correct SW, irrespective of the motivation for the model. And because of that, we can utilize the CFS distribution, which you may remember is a continuous distribution of non-interacting species Lamm equation solutions, to correctly determine the SW values of interacting systems, as long as the CFS gives a good fit of the experimental boundaries. In fact, we have shown that integrating the CFS distribution corresponds directly to this mass balance calculation. And this all holds true in the presence of chemical reactions. Now, it has been assumed in the past that the weight average S value so determined is obviously a signal weighted average of all species in solution at the plateau concentration. But this is true only strictly if signals are conserved in the chemical conversion between species. But in this case, we can carry out a concentration series of sedimentation experiments and fit the resulting isotherm of SW as a function of loading composition with a binding model. Now, finally, there is a fine point whether the relevant concentration for the isotherm modeling is the solution plateau or the loading concentrations. Both can differ a little bit due to the radial dilution in the sector-shaped cell by maybe on average 10 or 20%. But because one typically determines the SW values not from the two scans like sketched here, but from a large number, including early scans, one can argue that it should be an average that is close to the loading concentration. In practice, this makes an insignificant difference and is usually ignored because our errors in measuring protein concentrations are typically larger than the differences. Let's stick with this a little longer. Since we have said the weight average S values don't depend at all on the boundary shape, we can imagine a hypothetical boundary with the same S value that is infinitely sharp, like a single particle that does not diffuse. Non-diffusing particles are a time-honored model system for sedimentation processes. In fact, in the 1950s, Gilbert and Jenkins have developed their famous algorithms for solving Lamm equations of interacting systems in the approximation of non-diffusing particles. And to achieve that, they applied a transformation from space to a velocity axis and taking the limit of infinite time. I will come back to that later. Also, in the context of weight average S values, Robert Goldberg has introduced the concept of an infinitely sharp boundary already in 1953. Clearly, if the sharp boundary preserves the total areas under the curves, they will produce the same S value. Nowadays, we can express sedimentation of non-diffusing particles easily with the help of the Heaviside step function, but that wasn't available at that time. Okay, so how do we determine SW values in practice? Here is a set-fit window with loaded scans showing a fit with the CFS model after we have integrated the distribution. The integration will change the color of the fit function in the raw data plot to red for those portions of the scans that provide information on the integrated CFS peaks. At the same time, a little window will pop up and report integration limits, the total signal as well as the weight average S value. We can follow the same procedures for all experiments in the concentration series, which are usually data from cells in the different rotor positions of the same run. And then we can write a text file with a column for micromolar concentrations and a column with the SW value. That is what ZFAT reads for isotherm analysis. We will go through that in more detail shortly. Alternatively, once we have the CFS fit in ZFit, we can copy it over to Gussie, which is a very convenient utility program from Chad Braudigan. It can be used to create CFS overlays, make publication quality graphics, and carry out integration and creation of isotherms files for ZFAT. Here is how these isotherms of SW values as a function of concentration might look like, for example for heteroassociations. The concentration series are usually carried out in some rational way, like a serial dilution of stock mixers at some molar ratio, or a titration of a constant concentration of one reactant with increasing concentrations of the other. In that case, these curves are examples for how titration isotherms might differ for 1-to-1 and 2-to-1 interactions. However, it's not essential that the concentrations are picked in any such rational way. In any case, once we have a set of SW values as a function of concentration, we then fit it with a model that describes the signal-weighted average sedimentation velocity. The theoretical expressions will take a form similar to this, where we have contributions from each species with their respective signal contributions and their populations taking into account mass action law. Typically, we can run each component by itself so that we have S values and signal coefficients of both free species, leaving as unknowns just the binding constant and the sedimentation coefficient of the complex. We will see later that this might need to be extended by terms for hydrodynamic interactions, which are obligatory as we go to very high protein concentrations. And that's all there is to this. Let's recap. For this way of analyzing protein interactions, we do not need to care about the boundary shapes. It is valid equally for self-associations, heteroassociations, and independent of reaction kinetics. It does require a model for the interaction scheme, and then we can determine the binding constants from the fit. Also, in principle, we get an S value of the complex, which might be subject to interpretation with regard to implications of the shape of the complex, for example, considering structural hydrodynamic models. But how precise are the complex S values from this method? To answer this, let us look at the full binding isotherm. After all, for heteroassociation, it is a function of the two loading concentrations of the interacting components, both A and B. And this forms a surface in concentration space, and here this SW surface is plotted as a function of both concentrations in log units. The previous isotherms we've shown were an equimolar dilution series, here along this line, and the titration isotherms, which start at a constant of one concentration and keep that constant and then increase the concentration of the other. In any case, one does not have to adhere to any particular scheme, as I mentioned before, and you can probe this surface any way you want, as long as you sample the shape of the surface in some way. But what I want to point out now is how steep this peak is. And for the SW value to be close to the S value of the complex, to extrapolate that value and to estimate it, you need to be here on this ridge. And if we are not at least somewhat close, then the errors can get very large in our best fit values for the complex S value. We also have to consider that maybe our knowledge of the experimental concentrations is often not very accurate, and so that could add to the errors in our estimate of the complex S value. And when we look at this SW isotherm, it should not be forgotten that this is actually a signal-weighted quantity. These two isotherms here are for the same interacting systems. Just on the left, the first component has a much lower signal contribution than the second component, and on the right, it is the other way around. You can see how the shapes of the isotherms are different, especially around the peak. As a teaser, I will tell you that there is a specific path, the dashed line here, where these two isotherms are the same. Actually, these are concentration combinations where the system sediments as a single boundary. And this is a segue to the physics of the boundary structure. But before we go there, if you are watching this video as part of the workshop and have access to some of the example data, then this would be a good time to stop the video and practice with the provided data sets how to carry out isotherm analysis in ZFAT. So now we are ready to dive into the key question, what is really going on in these mysterious reaction boundaries? This will lead us to the concept of an effective sedimenting particle and then help us to develop a quantitative analysis that allows us to take advantage of the boundary structure. Here's the problem. Let's say we have an interacting system consisting of the green triangle binding the blue square to form a complex. We want to consider them locally in chemical equilibrium at all times, meaning they can rapidly associate and the complex dissociates all the time, and doing so they generate average populations of free and complex species as predicted by mass action law. Now imagine the triangle when it's free migrates slower than the square when it's free, but the complex is heavier and moves faster than either one. So how does the system sediment? We have seen that for whatever reason such a system can form at most two boundaries. And in terms of terminology, we call the fast boundary a reaction boundary and the slow boundary the undisturbed boundary. The reaction boundary is concentration dependent, whereas the undisturbed boundary migrates like one kind of the free species. For that reason, it has sometimes been mistaken as a molar excess of one component, but we'll see shortly why this is not true. Let's step back and consider all possibilities how one could envision such a system to sediment. Could it sediment all at once, with all components in sync, like here? That doesn't sound too crazy for me. It's certainly true if the individual molecules are in the complex that they'll sediment at the same rate, because they are physically coupled. But when they are free, they will migrate with different velocity and get out of sync, and our perfect imaginary boundary would start to unravel. Okay then, if they are not in sync, then there must be zones or volumes of the solution column that have different composition. And these zones are separated by moving boundaries because there are concentration steps that migrate. It is also clear that components in any zone cannot coexist without forming complexes because of mass-action law. And I think you will find it obvious also that the zones to the right must be the faster sedimenting ones, and so they must have more complexes and higher number of components. So could that be a possible species composition of zones for our AB system? No, since they couldn't coexist without forming complex here. Similarly, such a configuration wouldn't work because this mixture would sediment faster than the one to the right. But this one would definitely work, as would that. Now, this argument can be generalized, and we can conclude that if we had, for example, an n-component interaction, there would be at most n zones, the fastest one with a loading mixture, trailed by zones with consecutively smaller number of components and lower speed. Let's look at the zones in more detail. Dependent on the components that coexist in a particular zone and each total component concentration, mass action law dictates what the concentration of each individual species must be. So given the component concentration, we can track for each species how much of it is present in each zone, here for example 3A, and how much it changes across a zone boundary. And that change is how much that species will contribute to what we measure as sedimentation boundaries. And it is important that the concentration steps of all species migrate in sync with the zone boundary. Now we can package these fundamental considerations into a model of the sedimentation process. You will remember that we discussed earlier the idea of an infinitely sharp boundary that migrates, reflecting the sedimentation of non-diffusing species. And we found the very useful connection between the S value of that boundary and the integration of diffusion deconvoluted CFS distributions derived from experimental datasets. So what can we do with these step functions? Well, we can combine them to represent for each species the concentration step migrating with the zone boundaries. This, for example, is species I. Its change across the boundary B in concentration, and the boundary moves with the velocity SB. And this is an ansatz for the spatiotemporal description of the system, which we can insert into the Lamm equations of the system coupled with reaction fluxes, in the limit of no diffusion. Now, the nice property of these step functions is that their derivatives are Dirac delta functions, and those break the partial differential equation up into an algebraic equation system, which we can solve for the unknowns, which are the S values of the boundaries and the amplitudes of all species. And this can be done in a general way. I will spare you the details, which you can read up in the literature. It's been implemented in ZFAT, but it is interesting to ask why this was not discovered by the giants in the field in the 1950s, when there was such a tremendous interest in the development of AUC in physical chemistry, biochemistry, and molecular biology. I think it boils down to the simple fact that Heaviside step functions and Dirac delta functions were just not yet widely known. They were being developed at that time as tools for the emerging quantum electrodynamics. Without showing you the derivation, one of the most important results is this expression for the velocity of a zone boundary. We can identify this as the weight average S value of all species that contribute to the concentration step. And from that we can derive recursive rules for the boundary patterns. First, consider the loading mixture, which will be the composition of the fastest zone. We can easily calculate what is the time average or population average S value of each of the components in there. The fastest has to be the component that is entirely contained within that zone, and its S value is the S value of the zone boundary. The other components are slower in their time average, but we can subdivide those components and leave out some free species of that component such that the time average of the rest matches the S-value of the faster zone. What we need to match the time average S-value is the co-sedimenting fraction, and the rest is left behind and will constitute the next slower zone. And we'll do that subdivision for all components. And that will give us the composition of the slower zone. We can start over, use mass action law to calculate all species and all time average component S-values, and iteratively proceed until we have just a single component left, which then migrates with the S-value of that free species. Even though the derivation was based on the math, we can try to generate a mechanistic picture of the co-sedimentation. The molecules that constitute the undisturbed boundary are of course distributed throughout the entire solution column. But those molecules that happen to be located in the zone of the reacting mixture might get an extra boost occasionally because they are dragged along by the binding partners and get accelerated to faster momentary velocity when they are in the complex. Let's pool together those that got pulled along. We know how much that is. And we also know that all components co-sedimenting in the group have the same time average S-value. And so we can consider them forming a dynamic sedimenting unit, which we call the effective particle. Here is an animation of a single slice of the solution column, illustrating how the dynamic co-sedimentation works. Please disregard the vertical movement. That is just so that we can better track the individual molecules. Also, the theory is for infinitely fast reactions, but we have slowed down time so that we can see the individual binding events. Relative populations and velocities, however, in the cartoon are faithful to the underlying experimental parameters, which are shown here in relative units. There are a lot of squares that occasionally bind the slower moving triangles. Doing so, they create a faster sedimenting complex each time. Then they'll dissociate again. And that makes the triangle fall behind, but then another square comes along to pick it up. With this dance, they can all co-migrate in sync, macroscopically as if they were a discrete particle. In fact, I picked the total concentration of triangles and squares intentionally such that they precisely combine into this reaction boundary, and there is no undisturbed boundary. You can see that this effective particle macroscopically has a molar ratio of 2.4 and sediments with a velocity of 0.75, which is in between that of the faster component and the complex. The effective particle picture is really the key to sedimentation patterns of interacting systems. All of the mysteries we listed before can now be naturally explained. For example, we observed previously that forming more complex doesn't necessarily lead to a faster reaction boundary. To visualize this, let's repeat the same simulation but with fewer squares. If we have equimolar triangles and squares, then some of the slow triangles have sadly to be left out of the reaction boundary dance because they would drag down the average too much. After all, the average for the squares cannot be less than the velocity of the free squares. That's the lower limit. And therefore the triangles partition into those that contribute to the effective particle and those that create the undisturbed boundary. Now compare that with a case where we have twice as many squares, and everything else is the same. Now that's down here with twice as many squares. We can let them start at the same time and see who wins the race. You can already see that the higher concentration loses. Yes, we do have much more complex, but also more free squares in this reaction boundary. And those will keep the average velocity of the reaction boundary lower, and that's why it arrives later. And this behavior of slower reaction boundary with increasing squares continues until we exceed a critical concentration, which is the magic concentration that we looked at before when everything matches up to a single effective particle. Now these are the simulations we just looked at. If we increase the number of squares beyond that magic point, then two things happen. For one, the average velocity of all squares drops since there are so many free squares. But also, from mass action law, the triangles are more frequently in the complex state and their average goes up. In fact, they will now govern the velocity of the effective particle, and some of the squares have to be left out. So now the squares form the undisturbed boundary. And the more we drive the triangles into the complex with the excess squares, the faster the reaction boundary will be. Let me start the race and see how this plays out. Now in the bottom panel you can see that the triangles are almost always in the complex state. And that makes them move very fast, while a lot of the squares have to be left behind. Now this is maybe a little bit crazy, and you may want to rewind the video to observe in detail the dance of the effective particles in the different conditions. Alternatively, you may start up ZFAT and switch to the interaction model, enter molecular parameters of your choice, and then start up the Effective Particle Explorer tool. This will show you the isotherms of different properties, and here, for example, it shows the reaction boundary S value in a heat map as a function of log of concentration of components. In the software, you can click at some point in the isotherm and an animation will pop up that reflects the effective particle at the particular concentration. I find this visualization very useful to get some intuition of reaction boundaries. For simple interacting systems of two components, the effective particle theory leads to closed analytical expressions for the reaction boundary S value, its amplitude, and its composition, which describe the entire concentration space at once. And this is how the isotherm of reaction boundary S value looks like as a function of concentration of components. The one that sediments slower than free, which is called A, and the one that sediments faster than free, which we call B, on a log scale. And what we have just examined in the animations is a titration series of constant A and increasing B. For general n-component systems, there is no closed-form analytical solutions. But we can solve the algebraic equation system easily enough. One very interesting feature is that the equations break apart into different cases. And ultimately this is because the reaction boundary cannot sediment slower than the free form of the faster of the two components. Now, all points in this line describe where the loading concentrations are just perfectly combining to produce effective particles without any undisturbed boundary. Above and below this line, the nature of the undisturbed boundary changes. We call this line a phase transition. It is very clear that this line is not symmetric with respect to A and B, and that is because you need to keep the slower sedimenting component in a complex a much larger fraction of the time for it to be entirely in the reaction boundary. Therefore, the asymmetry increases the more the S value of free A and free B differ, which is illustrated down here, showing the phase transition line for different size molecules. And this explains another of the list of mysteries of boundary patterns, which was that the undisturbed boundary switches at concentrations that do not reflect complex stoichiometry. Let's push these ideas a little further. So far, we have used a diffusion-free approximation to predict the overall boundary structure, their velocities and composition. But since it predicts the concentration steps, can we use this to estimate the expected diffusion coefficients? If we look at the diffusion-broadened reaction boundary, the concentration gradients of all free and complex molecules are fairly constant across. And so the question is, having predicted the size of these steps, will a single gradient average diffusion coefficient of all species be a useful quantity? And to test this, we can simulate full Lamm equation solutions of our interacting system, as always add some random noise to mimic experimental data, and then we model this with a C of S distribution to extract a single empirical diffusion coefficient for each peak. And then we compare this with a prediction from effective particle theory for the simulated conditions. And when we carry this out for different concentrations and different assumptions on diffusion coefficients, then we can find that the predictions very nicely describe the empirical values. There is no adjustable parameter in these comparisons. So I'd like to conclude that not only do the reaction boundaries sediment like effective particles, but they also approximately diffuse like you would expect of real particles. And this can give us an idea not only of the reaction boundary S value, but also the boundary shape. I want to talk about another second-order effect causing concentration and composition gradients within the reaction boundary. To explore this, we assume a little bit more detailed view than the one we used to introduce effective particle picture. So let's look at the cartoon of the reaction boundary here of mixed species and pretend that we have a microscope to observe the individual molecules. And imagine there's a demon that has the power to stop time and alternate sedimentation with chemical reactions. Since complexes momentarily sediment fastest, they will clear some solution of the complexes. And now let's freeze sedimentation and let chemical reaction happen. You observe the concentration and composition in this region is lower, and when it re-equilibrates, it'll again have to form some complex, although probably a little less than original. Now freeze the reaction and let sedimentation resume. The newly formed complex will again sediment, and once again it lowers the concentration and alters the composition behind. And as this goes on, we arrive at a concentration gradient, even though we have not talked about diffusion. And in the time-honored transformation of the radial to velocity axis, we'll see how this concentration gradient actually transforms into polydispersity in sedimentation velocity. And this is a consequence of the changing composition within the reaction boundary. Now, this picture is actually not so different from the computational strategy developed by Gilbert and Jenkins in the 1950s and 60s for sedimentation coefficient distributions of diffusion-free, rapidly interacting systems. They calculated these structures, which were called asymptotic boundaries. And it is important to mention that the overall pattern and S values are highly consistent with our effective particle model. But they provide a shape and they have a polydispersity to them, which constitutes a second-order effect. Although, mostly, they'll produce fairly narrow distributions, except in situations that are close to the phase transition point, when the concentration gradients are maximal. So, in summary, we find that the reaction boundaries sediment like effective particles, diffuse like effective particles, and are slightly heterogeneous. Now, the good news is that our diffusion deconvoluted CFS distributions are perfectly suited to accommodate such polydispersity. What we learned from this Gilbert-Jenkins viewpoint is that there can be substructure and non-symmetric peaks, maybe exhibiting some shoulders, and we know where it comes from. But since our goal is not to interpret the details of the boundary shape, and not the shapes of the CFS peaks, we can simply integrate the relevant peaks and get the average quantities that we need to model with effective particle theory. Okay, now we have a theory for the boundary structure of interacting systems, along with a method to extract from experimental data the relevant quantities for this theory to be applied to. How well does this work? You may remember our experiments titrating the killer cell receptor Ly49C with MH2+1 molecules at increasing concentrations. Now, with the effective particle theory, we can globally model the S value of the reaction boundary, which is always the fastest of the two visible boundaries, and also the ratio of the boundary amplitude of the reaction boundary to the undisturbed boundary, and of course the traditional overall weight average S value. The data points are extracted from integration of the CFS fits. Here, S fast refers to the reaction boundary S values, these SW are the weight average S values, and these are the amplitudes of the reaction boundary and the undisturbed boundary. As you can see, the best fit with effective particle theory is very nice, and we arrive at a consistent parameter set as we did with the Lamm equation solution fit. Essentially, we can now extract binding constants from the bimodal structure of the reaction boundary, but without the necessity to worry about the detailed diffusional broadening or kinetics, and with the ability to exclude preparative impurities that may show in minor unrelated CFS peaks that we do not integrate. We can also try the imposter 1-to-1 model, which would give this best fit and clearly does not fit. Another good way to examine the performance of EPT, or effective particle theory, is to generate sedimentation patterns solving the full set of Lamm partial differential equations, adding some noise to the simulated boundary data, calculate a CFS distribution from this, and then see how the peaks line up with the EPT predictions. Let's do this for our 3-component test case that we discussed previously, where we had a bispecific antibody binding 2 different ligands, forming 6 species in solution and exhibiting up to 3 boundaries. This system depends on 3 loading concentrations, and here's a graphical representation of the isotherm of the reaction boundary S values, which is color-coded in this volume. It's a function of log concentration A, log concentration B, log concentration C. What we had explored previously were 2 titration isotherms along this path at constant C. And here you can see the sedimentation coefficient distributions representing the simulated boundary patterns. It's the solid lines. And you may remember they showed fast boundaries monotonically increasing in one case, and in the other case decreasing, then splitting up and then increasing. So the symbols here are the predictions from the effective particle theory. And this is a little hard to compare, but it's easier if we turn this into a cumulative sedimentation coefficient distribution. And I believe you will agree this nicely matches. Of course, the C of S distributions have a finite width, whereas the effective particle theory is, of course, discrete, but this will not matter in the isotherm analysis. And for this system, I want to point out how information-rich this analysis is. For one particular loading mixture, the C of S profile is shown here, and the pie charts reflect which of the 6 species contributes to that particular boundary. You can imagine that if we used specific detection, maybe with labels or just based on different extinction coefficients in the UV, we get multiple C of S with varying signal amplitudes. And this plot is an illustration of the S values of the 3 boundaries we detect for this 3-component system in our titration of A into constant B with a constant C. The undisturbed boundary is in magenta, the S values are here, and this is the concentration of A. The 2-component boundary is in blue, and the reaction boundary that is fastest is here in black. I have drawn the width of these lines to reflect the relative amplitudes of these boundaries. And just to contrast this, the dashed line shows the weighted average S value as a function of concentration. And this plot illustrates very nicely the information gain that we have once we are in a position to analyze the entire boundary pattern. And this can set us up to study much more complex interacting systems. Furthermore, such isotherms of reaction boundary velocities and SW values can be incorporated into global multi-method modeling in combination with data from other techniques. This is a topic for a different video in this series. This was a long section, but now we've done the hard work. We've demystified the reaction boundaries. Through math and physics, we have arrived at simple rules that center around the time average velocity of the molecules. Effective particle theory can explain the mechanism of cotransport, and in this way predict the entire boundary pattern, which include the boundary number, their S values, amplitudes, and compositions, for any rapidly interacting system. Importantly, to apply this, we do not have to fit Lamm equation solutions, but instead we can focus on integrated C of S peaks of interest. And as a method that relies on having mixtures of purified overexpressed protein samples, this is critical because it allows us to deal with systems with imperfect purity, and that will set us up for studying multi-component mixtures. So in the next section, we'll talk more about how to apply this in practice. And this will assume that you are very familiar with how to set up sedimentation velocity runs in the AUC, including the use of different optical systems and their detection limits. And likewise, we assume that you are very comfortable in carrying out basic CFS analysis. The analysis rests on a series of sedimentation velocity data from samples at different concentrations. If you have an idea where the equilibrium dissociation constant might be, then ideally we would want to cover a range from 10-fold below to 10-fold above Kd, and even larger if possible. It is a good idea to acquire multiple signals. You will see more about this later in the section on multi-signal analysis, but even for isotherm analysis of single components, switching detection systems allows us to cover a larger concentration range. And this can be leveraged further by using sample cells with different optical path lengths. And then we carry out a C of S analysis of all the mixtures, copy the distributions and look at the overlay. You can do that in Excel. I have a little MATLAB script, or often the most convenient approach is GAUSSI. And Like we have discussed earlier in this video, we can classify whether the reaction is slow or fast on the timescale of sedimentation. If the chemical exchange is slow, then we usually hydrodynamically resolve all species, and we get CFS peaks that do not change in S value, but only exhibit different heights corresponding to the shift in populations for the different mixtures. When we recognize we are dealing with a slowly equilibrating system, then usually we double-check that the incubation time of the sample prior to the run was sufficient. Sometimes we find ourselves repeating the same experiment in different configurations, for example, reaching the same sample concentrations through dilution of highly concentrated equilibrated stock in comparison to another sample that was directly mixed to the same final concentrations. We should get the same result if the samples were sufficiently equilibrated. For systems with fast kinetics, it is no surprise anymore that we have reaction boundaries with concentration-dependent S values, and maybe even with some substructure, usually in addition to some undisturbed boundary. Seeing these overlays often makes us think about the reaction scheme and the S values of the complexes. We will see later how multi-signal analysis can greatly help in clarifying the stoichiometry. In the case of slow kinetics, we can read the complex S value basically from the location of the complex peak. But for fast system, of course, we are looking at effective particles, and the complex S value might be much larger than the S value of the fastest sedimenting peak, depending on condition. Now we are ready to construct the isotherm file. Here's how to do this from scratch. We start for each concentration in our ZFIT window showing the CFS analysis. And then we use the integration tool and integrate around the reaction boundary. ZFIT will report the S value as well as the signal amplitudes of the integrated peak, and we will want to write this down. And then we can do the same thing for the other peak to get its amplitude. And of course, we want to integrate the entire distribution to get the weighted average S value. So we do this for the entire concentration series and construct a standard ASCII file, for example using the notepad with the extension .isotherm. The contents is a matrix of numbers where the rows correspond to the different experiments and the first columns correspond to the component micromolar concentrations. There will be one column for self-association and two columns for two component systems and so forth. The last column is, in this case, the S value, for example, the weighted average S value, or it might be the reaction boundary S value. So we create these different files for the different types of features and load it in ZFAT. We load it as a new ZFAT experiment of the sedimentation velocity isotherm type. We have to specify whether it is the SW isotherm, the reaction boundary isotherm, which is called EPT SW fast, or the signal amplitudes of the undisturbed and reaction boundaries, which is EPT partial concentration. And this was assuming that you have a fast system. If it is slow, then you can load the isotherms of species amplitudes called partial concentrations, and that will be modeled directly with mass action law. Either way, it is very important to enter the extinction coefficients for each component and the optical path length. And since you can load many different isotherm files at once, the entry of the different extinction coefficient provides an opportunity to carry out global analysis and take advantage of differences in signal contribution. As I have mentioned before, Chad Browdingham's SCSI is a convenient software to inspect the CFS overlay, carry out consistent integrations, and export the requisite isotherm files for ZFAT analysis. But when we look at the overlays, maybe we have trouble sometimes identifying different peaks. For example, there might be a complicated pattern with multiple boundaries. We are not sure which is which. Or because of low signal-noise ratio at lower concentrations, the regularization in CFS makes the boundaries merge together. To some extent we can modulate this by changing the regularization p-value, or even use Bayesian regularization, for example assigning the expected S value of the undisturbed boundary as a prior. But at some point this won't help anymore. In this case there's a method to rescue we call sedimentation boundary pattern analysis. Basically, we can just integrate our CFS distribution, trying as best as we can not to divide peaks, and then we just write down the limits of integration that we happen to use. Then we let the software do the work of simply adding up all the boundaries that fall within that S value range. And this is how this will look like in ZFAT, where we have added a feature of boundary pattern isotherm. We will have to tell the software whether we are looking at S values or signal amplitudes, and what the integration range was. And one decision we have to make is whether the reaction is slow or fast. And then ZVAT will be able to model this either with effective particle theory or simple mass action law, as the case may be. Since this alleviates the need for us to resolve and identify all boundaries, it makes life much simpler to study more complicated interacting systems and still take advantage of this structure that we see. And with this in mind, we have created a model editor in ZVAT that lets the user specify the reaction scheme for up to 4 components and any number of complexes. So generally, we would load multiple isotherm files from different concentration series, different signals, and different integration ranges. And here is an example of such an analysis. You can see we have acquired a lot of data for a system with multiple complexes. Here's another analysis, and you may recognize that there's also an isothermal titration calorimetry dataset hiding in here, which is just a teaser for our global multi-method analysis. So, and if you want to exercise a little bit, then you can find these datasets, which we'll try to make available through our website. I have mentioned already several times before the opportunity to detect the sedimentation process with different optical systems. And in the context discussed so far, we would just generate separate CFS distributions from the different signals and separate binding isotherms, and then join them in a global analysis in the end. But there's also an opportunity to generate multi-component sedimentation coefficient distributions up front. And this can be particularly powerful when we can hydrodynamically resolve the complexes. And that's certainly the case for slowly interacting systems, but even reaction boundaries of fast systems can in many cases provide good approximations of the complex stoichiometries. So let's see how these multi-component sedimentation coefficient distributions work. When we introduced the CFS analysis, one distinct conceptual difference to historic approaches for sedimentation coefficient distributions is that those were based on approximate data transformations, whereas the CFS method is an explicit boundary model. And this is the reason why it can provide diffusional deconvolution, and, as we soon realized, it sets the stage for fitting multiple experimental datasets at once. And so, not long after introducing the CFS method for an ordinary sedimentation boundary dataset, we developed an approach to globally model data acquired from the same cell with different optical systems. As we've shown later, there is a similar opportunity to carry out global analysis of SV data from multiple rotor speeds and gravitational sweep sedimentation for very broad size distributions, or to global analysis of sedimentation data acquired in solvents of different density in a density variation approach to determine partial specific volumes. But in the context of sedimentation velocity for studying interacting systems, the variation of the data acquisition signals is particularly useful. The experimental basis is that we can observe the sedimentation process with different optical systems quasi-simultaneously, and that gives us the opportunity to spectrally distinguish different sedimenting components. For example, proteins have different fractions of aromatic amino acids, especially tryptophan, and as a result, the ratio of refractive index signal to UV absorbance signal at 280 nanometers can be very different. Furthermore, our proteins of interest often have different ratio of tryptophan to tyrosine, and as you can see here, that'll affect very much the ratio of the signal at 280 relative to the minimum around 250. And of course, if we are out of luck, we can attach chromophoric labels if need be. If we are studying interactions between proteins and other macromolecules like nucleic acids or carbohydrates, then there will be naturally a huge difference in spectral properties. In terms of practical application, we have currently implemented multi-signal CFS for absorbance signals from up to 3 wavelengths and/or refractive index signals. As you will see, the increased data acquisition time is not critical, since we are measuring the progress of the boundary with each of the signals, and therefore we don't lose much spatial information by acquiring different signals. We actually may gain if one of the signals is Rayleigh interference, which is very quick to collect. This is going to be the only math slide on this. Previously, we have modeled our experimental data as a linear superposition of Lamm equation solutions, with the coefficients representing the sedimentation coefficient distribution. In the multi-signal context, we extend this to all data at each wavelength lambda. And we allow for the fact that there are different components k, each of which has its own sedimentation coefficient distribution Ck of S. We need to know what is the signal contribution of each kind of component to each of the signals, which is denoted with the extinction coefficient matrix. One quick remark on the terminology. The refractive index sensitive signal from the Rayleigh optics is mathematically not any different from other absorbance signals at any wavelengths. And therefore I might refer to an extinction coefficient when I really mean an interference fringe increment. And this doesn't make any difference. It will be useful to express extinction coefficients in molar units so that the resulting C of S distributions also are in molar units and their ratios directly reflect component stoichiometries. Let me give you some examples. A first experimental test of this concept was mixing two non-interacting proteins, IgG and aldolase, which have similar molecular weights but different extinction coefficients. Here are the raw data with interference optics and absorbance at 280 nanometers and 250 nanometers. All of them show a single broad boundary, and a conventional CFS analysis of each data shows only a single broad peak for each. So in the multi-signal CFS, we are asking to get the contribution that each component is making to each of the data sets. Importantly, we get a great global fit. And also, we get component Ck of S distributions calculated with the spectral signature of each protein. And here is the distribution of species that look like aldolase. And this is the one that has IgG's spectral signature. Now you can see that our broad C of S peak can be decomposed into contributions from two spectrally distinguishable species. The dashed lines here show the C of S distributions we get running the controls containing only aldolase or IgG. Also, these controls were used to determine the relative extinction coefficients for each component. So it turns out that our ability to spectrally distinguish these two proteins has compensated for the lack of hydrodynamic resolution. You might say we collected three signals, and so we should be able to distinguish three spectrally different proteins. And thinking along this line, we threw BSA into the mix. The raw data at each signal looks like two boundaries that are present since BSA sediments a bit slower. But looking at the component distributions from multi-signal CFS analysis, we can see that there are three species, aldolase, IgG, and BSA. Again, we resolve the IgG from the aldolase on the background of additional BSA. Why does this work so well? It is because we have tens of thousands of data points with fairly high signal-to-noise ratio, and the boundary model can extract this information. Obviously, another prerequisite for this to work is that the signal contributions need to be linearly independent. But here's the thing. If we have hydrodynamic resolution, then this can help us out to discriminate components. So if we could separate ranges of hydrodynamically distinct species, then only within such a range do we require spectral resolution. And in this way, we can introduce the notion that spectral decomposition could potentially be different in different S ranges. So just to practically manage such a concept, we might, for example, add up spectral properties according to some stoichiometries. For example, we can say that in a certain S range, a component kappa has a spectral signature that is the sum of certain basic components K. You can see where this is going. If we have protein complexes, barring any hypo or hyperchromicity, then their extinction coefficients are the sum of their individual components. And in the lingo of ZFAT parameters, these would correspond to what's called chromophores. Let's put these ideas together in an S-value line. For very small S values, we would probably not expect our proteins to contribute at all because they sediment faster. Nonetheless, in practice, we may have sedimenting signal contributions, for example from unmatched buffer components contributing to the refractive index signals, or perhaps some additives like DDT or nucleotides contributing to absorbance signals. As we are really not interested in studying these in detail, we might just span the spectral space in this low S range independently with components contributing exclusively and independently to these signals. But then, for larger S values, we are above the range of buffer contributions and in the range where the free proteins might sediment. In this range, we might span the spectral range with the properties of the independent components. And if we know from controls where our separate proteins sediment, then we can conclude that in the S-value range above, anything we see has to be at least a one-to-one complex or a complex with higher stoichiometry. So in this S range, we could constrain the spectral space to some linear combination of the component spectra. Remember, our multi-signal C of S is a model that we construct, and we can embed constraints and test hypotheses if we wish. You may remember our earlier example of PLC gamma and SLP76 peptides sedimenting in a mixture, which was observed by absorbance at 280 nanometers and also with interference optics. Previously, I showed you how it is possible to model this data nicely as a Lamm equation solution for an interacting system where the two proteins form a slowly dissociating one-to-one complex. But maybe we are not sure about the model. So we can use the multi-signal CFS analysis and find the component distribution of PLC gamma and SLP76 separately. Here, the spectral decomposition in the peptides was applied for all S values above 0.4 S. And below this threshold, we added discrete species representing just interference and absorbance contributions separately. Clearly, there is an excess of buffer signal, which we can see here in the interference data. And in the analysis, this comes out to be this species, which probably just represents the sodium chloride that is not optically matched. But nicely, from the multi-signal analysis, we can recognize the formation of a complex at 1.1 S, where PLC gamma and SLP76 co-sediment in what seems to be a one-to-one complex. Just like it is illustrated here, we can then ask further if we can constrain the S-value range above 0.8 S to one-to-one and higher complexes. That shows us clearly that there is a majority of one-to-one complex and perhaps only traces of species with higher stoichiometry. It is worth pointing out that just from the spectral composition of this peak, we cannot distinguish whether it's a one-to-one or two-to-two complex. However, here the hydrodynamic information comes to rescue, since the mass of a two-to-two complex would usually be associated with a much larger sedimentation coefficient. And so this is an example for how you can construct the multi-signal analysis with different spectral constraints that match what we know or hypothesize about the sample. Now that we have a convenient way to spectrally measure the molar ratio of a complex, does this require for complexes to be long-lived? Or will this work too for rapidly interacting systems? Again, effective particle theory can help us understand this. Let's go back to our previous example of moderately sized proteins with somewhat dissimilar free S-value, forming a one-to-one complex in rapid association-dissociation equilibrium. Here is a two-dimensional isotherm with a heat map indicating the molar ratio of the reaction boundary, which is what we could measure directly with the multi-signal CFS method. Where it is red, that's where the composition is 0.8 or higher. As you can see, as long as we are about threefold above Kd with either one of the components, but maybe better not both at the same time, the reaction boundary will reflect the complex stoichiometry pretty well. But that's not always the case. Let's assume there's a much greater discrepancy in the free velocities. In that case, we can still get away with threefold Kd of the smaller component, but the molar axis of the larger component would have to be way greater. On the other hand, if the free components sediment with similar S values, then we are okay at almost any case, even at a fraction of Kd. It's very interesting that we don't even have to populate the complex much in this case to measure its stoichiometry. But the bottom line is that it will always be good to have an idea where the Kd is and then go with an excess of the smaller components. For any particular system, we can consult the EPT Explorer tool in ZFAT to generate the composition isotherm, like here, and then the movies to rationalize what's going on. Let us look at examples. You have already seen this one, so now let's look at two others. Here is an antibody D1.3 binding to HEN-EC lysozyme. The lysozyme by itself sediments at just below 2S and the antibody at about 6.5S. They have very different spectral contributions to interference and absorbance at 280 nanometers. And using the predetermined extinction coefficients from control experiments, we can analyze the mixture with lysozyme in excess over antibody. And the solid lines show the component sedimentation coefficient distributions in red for lysozyme and in blue for the antibody, in molar units. You can see, for one, that the S value has shifted. Clearly complexes have formed. And from integrating over the two peaks, we can confirm that there's about twice as much lysozyme than antibody in the complex, which therefore confirms the expected 2-to-1 stoichiometry. Here is a sketch from one of our first applications to adapter proteins of the immunological synapse in collaboration with Larry Samuelson's lab. I don't want to go into the detail, but you might glimpse that we used 3-component multisignal sedimentation velocity analysis to establish the kind and stoichiometry of complexes that form under different conditions. In this case, different phosphorylation status and multivalency of one of the binding partners. Without going into any kind of detail, you can also visually recognize right away that there are multiple complexes that coexist. In practice, when is it promising to do a multisignal experiment? Usually, this is a good idea when studying multi-component mixtures, not only protein-protein interactions, but also protein-nucleic acid interactions or interactions of other macromolecules. It can be a very direct method to determine the stoichiometry of protein complexes and is particularly useful when there are multiple coexisting complexes, potentially in a system with unknown assembly mechanisms. Frequently, we can get away without any labeling, and it's possible to predict that upfront. The key are the extinction coefficients of all components at the signals we are planning to acquire. It is useful to arrange them in a matrix like this. Here, for example, we assume that we have two proteins A and B, and we are planning to acquire interference data and absorbance at 280 nanometers. If there is no significant modification to the protein, then we can predict both refractive index increments and the extinction coefficients in absorbance from the amino acid composition. And you will find a Z-fit calculator function which can conveniently provide these molar signal increments given the amino acid sequence. These extinction profiles need to be linearly independent, and this can be visualized geometrically in a plane where the signal contributions for each protein is represented as a vector. The spectral signature of the two proteins being independent means that the area of this parallelogram must be different from 0. The area can be calculated by the determinant of the matrix, and if we normalize this by the product of the vector lengths, we get a quantity called d-norm. Through a series of simulations, Shea, Patrick and Chad Broughtigam have shown that a d-norm value greater than 0.05 is usually sufficient to distinguish two proteins spectrally in a multisignal sedimentation velocity experiment. Maybe you are asking, what is the precision for determining molar ratios in a complex? Will we be able to discriminate a 2-to-1 from a 3-to-1 complex? To answer that, we can carry out a more detailed analysis of the error propagation from relative noise in the acquired data into relative uncertainty of component concentration. And this will depend on the number of meaningful data points, and conveniently there is a Z-fit calculator function that will predict all of that given the planned run conditions. Finally, there is an approach for using mass conservation as a constraint in the analysis. This can greatly help the spectral assignment. After all, we usually know how much of each component we load into the mixture, and that factors into the assignment of components to the sedimentation boundaries. You can find more details about this in this reference. So how do we run a multisignal experiment in practice? As we have already discussed, we need extinction coefficients for each component. Critical are the signal ratios, and they need to be determined experimentally. The predictions are not good enough. But one of the extinction coefficients must be known a priori. Often, for unmodified proteins, the refractive index signal increment is the most precise and straightforward to use. Then, in the multisignal experiment, we run all individual components separately, side by side with the mixtures, and we scan all the cells at all the wavelengths. For rapidly interacting systems, a good general strategy is to keep the smaller component in excess to saturate the reaction boundary. Of course, all signals need to be within the detection range, and it often helps to use different optical path length cells for different samples. Especially for the individual controls, we need to ensure that there is a good signal-noise ratio at all signals. And this is because we analyze these data first, fix one extinction coefficient and optimize the other. And that establishes the extinction coefficient matrix that will be kept fixed and the basis for analyzing the mixtures. And once again, here's the cue for going through some practical exercises if you are participating in the workshop. The last segment of this video deals with the unavoidable physical interactions between macromolecules that are based on the fact that they occupy finite volume and are sedimenting in viscous solvents. Now, these interactions will scale with the total concentration and as such will be particularly relevant when we investigate weak interactions which require high total protein concentrations. It has long been known and can be readily observed that inert particles sediment slower at higher concentrations. Without being specific of what's going on in this concentration dependence, just like we can expand functions in a Taylor series, we may write the sedimentation coefficient with a first-order term to describe the concentration dependence in the dilute regime with a coefficient Ks. In this context, it will be useful to think in terms of weight concentrations. Similarly, we can expand the diffusion coefficient. So far, this is just a phenomenological coefficient similar to virial coefficients, which we can measure. So, for example, for BSA, it is about 0.01 per mg per mil, meaning the S values will be reduced by about 10% at 10 mg per mil of protein. There is a huge body of work in the statistical fluid mechanics literature filling these coefficients with physical meaning. In 1972, Batchelor has published a seminal paper where he worked out the hydrodynamic interactions in a suspension of spheres and calculated a concentration dependence that is 6.55 times their occupied volume fraction. The two major contributors are solvent backflow and pair distance distribution. What is backflow? You may remember from the introduction to sedimentation velocity the visualization of the hydrodynamic flow field surrounding a particle moving through a viscous solution. The critical point was that the flow field decays only very slowly and quite a bit of fluid is dragged along with the particle, here highlighting the region where the fluid velocity is at least 1/3 of that of the particle. Let me illustrate what that means for different protein concentrations. This shows a cube of 20-fold particle diameter, each particle with a red highlight for the region where a solution is dragged along with more than 1/3 of the particle velocity. At 1 mg per mil, there is a good distance between the molecules and their flow field. At 10 mg per mil, it's already getting crowded. And at 100 mg per mil, the entire solution volume is red. Obviously, since hydrodynamic interaction is long range, motion of any particle is coupled to motion of all others. Let's go back to sedimentation. Since the sedimentation takes place in a fixed container, since there's volume of particles and dragged solvent flowing down, there must be solvent flowing up. And since sedimentation takes place against this backflow of solvent, this creates additional hydrodynamic friction and lowers the sedimentation velocity. And it's easy to see that the magnitude of this backflow effect grows with more particles sedimenting. Also, it becomes apparent why the relevant concentration scale is the hydrodynamic volume. We can approximate this with a hydrodynamically equivalent sphere, which leads to an inflation of the excluded solvent volume by approximately the cubed frictional ratio. The role of the particle distance distribution is also easy to see if you consider the probability that just randomly particles are in close vicinity. Here is a simulation of random placements of particles on a grid. Without any interaction, there is a distinct chance that particles are close by, here highlighted in red. If that happens, then they are in their mutual flow field and will be able to sediment faster jointly. As Batchelor has shown, we can attribute approximately half of the nonideality to depend on this effect. In fact, it is possible to express the particle distance distribution as a ratio of the second virial coefficient to the second virial coefficient of a hard sphere. And I should mention that the second virial coefficient can also be calculated in the framework of these phenomenological coefficients Ks and Kd, but since Kd is typically a bit difficult to determine reliably, this relationship is actually more useful in my mind. If there are attractive interactions where the virial coefficient is smaller, then they will be more likely to be close by and have hydrodynamic interactions, like in this case. Vice versa, if there are repulsive interactions, then they will on average keep greater distance so that non-ideality becomes larger. Taken together, this provides the link between the Ks values, frictional coefficients and virial coefficients, which we have tested and found useful, for example in the study of formulation conditions of therapeutic proteins. Essentially, this is a way to measure interactions even if they just modulate the probability that molecules are close by in solution, and even in repulsive cases. Because sedimentation boundaries exhibit concentration gradients, not all molecules will experience the same non-ideality, since they are in different solution volumes with different local concentrations. How does this impact the sedimentation boundary shape? To demonstrate this, I have simulated non-ideal sedimentation, and these are the boundary profiles at different points in time. They are for ideal sedimentation in the dotted line, and for non-ideality approximately like BSA, shown by the dashed lines, and then with twice the non-ideality coefficient, that's the solid lines. At low concentrations up here, there is little difference. But as we load 5 or 10 mg/ml, you can see distinct retardation of the boundary. And also, they get much steeper. And this is because the leading edge is at high concentration and migrates slower, whereas the trailing part is much more dilute and therefore migrating faster, and it can catch up. In fact, this sharpening can completely counteract diffusional broadening and produce a time-independent boundary shape in the limit of high concentrations or strong non-ideality. Since the boundary shapes will not conform to the standard ideal Lamm equation solutions, ordinary CFS analysis, for example, will not give a good fit and will likely arrive at unrealistically high frictional ratio estimates. And this is actually a telltale for non-ideality. However, weight average S values, as we have seen before, depend on the mass transport across a line in the solution plateau and therefore provide a good avenue to measure the non-ideality coefficient. For example, we could plot the S value as a function of concentration and the slope will provide Ks. This might look like this, here showing the measured SW values for the NIST map at different concentrations in buffers with different ionic strength. This is an example for how electrostatic repulsive interactions can impact the non-ideality. You might think perhaps non-ideality is not so critical if we are just trying to determine relative concentrations by measuring boundary heights in multi-component mixtures, and if we are not interested in precise S values or boundary shapes. But this would be a big mistake. Let's look at what happens if we have, for example, a mixture of 3 species sedimenting at distinct S values, each present at 10 mg/ml. Here are simulated sedimentation boundaries for convenience in mg/ml units. That would be what's measured, for example, with refractive index sensitive interference optics. As you can see, there sure are 3 different species sedimenting, but not all with boundary heights corresponding to their 10 mg/ml loading concentration. What is going on? Here are plotted the concentration profiles if we could follow each species separately in the sedimenting mixture. The slowest species is the solid line, the middle species are dashed, and the fastest sedimenting one is dotted. Let me walk through the first point in time in blue. The fastest species is entirely within the loading mixture and sediments exhibiting a normal non-ideal boundary. The middle species, the dashed blue line, is for the most part covering the same volume as the fastest species, and in that zone it will be retarded just as much from the backflow. However, a portion of that, since it's slower than the first species, will be in a region depleted from the first. And as a result, there's a lower total concentration here, meaning less backflow and not as much retardation. Therefore, the molecules of the second species in this region here will sediment faster than the molecules from the same species in the zone ahead. As a consequence, they will pile up behind the boundary of the first species. The same thing happens to the slowest species, being much faster in the partially depleted solution zones and therefore piling up. Usually we will not measure the species separately and might not observe the piling up directly, but in their sum it'll lead to masking of amplitudes of the faster sedimenting boundaries. And this is called the Johnston-Ogston effect. So how can we analyze interacting systems in the presence of non-ideality? As we have seen above, one theoretically appealing approach is to directly fit the data with systems of Lamm equations. However, non-ideal systems inherit all the disadvantages we've discussed before, chiefly the susceptibility to experimental imperfections and the dependence on parameters that might be ill-defined. For non-ideal systems, this is exacerbated due to the additional dependence on the non-ideality coefficients Ks, which strictly should be mutual interaction coefficients. I believe reliable analysis with this approach will be possible only in exceptional circumstances. Again, we can step back on the detail and just use mass balance analysis to model the isotherm of weight average S value as a function of concentration. Strictly, this takes such a form, considering the weighted contributions of all species and their mutual non-ideality. However, it is usually an excellent approximation in standard practice to describe the non-ideality with a single average coefficient. Ultimately, that's saying that the backflow in a mixture won't depend much on the identity of the protein. Such isotherms are illustrated here for a non-ideal monomer-dimer system. Shown are the measured weight average S values relative to the ideal monomer S value at infinite dilution, assuming a non-ideality coefficient of 0.01 per mg/ml and plotted against the log of total concentration. The dotted lines are the ideal isotherms, and you can see the effect of non-ideality in the solid lines, reducing the observed S values. In some cases, if we measure across a sufficient concentration range, we may be able to determine both Kd and the non-ideality coefficient. Since we'll not be able to get close in SW to the complex S value, there can be correlations between Kd, Ks, and the complex S value. It can be helpful to make assumptions constraining Ks, the complex S values, or both. In fact, there can be situations where the increase in SW is completely masked by the decrease from non-ideality. In that case, we will need to rely on estimated non-ideality parameters and complex S values in order to get an estimate for the Kd. One important point in all of this is to keep in mind that non-ideality depends on the total concentration. If there are other components sedimenting that are perhaps not detected, for example, if we use selective fluorescence and absorbance systems, then we may not be able to carry out a quantitative analysis since we are missing information on the local total concentration. Similar problems can occur if some solution components are presumed to be present in a loading mixture, but they rapidly sediment such that the observed sedimentation process actually lacks this component. This is one of several reasons why I prefer to carry out studies of non-ideal systems with the interference optics, since it can visualize all sedimenting components and we have a firm handle on the total concentration. For ideal systems, we have seen how the sedimentation coefficient distribution C of S can be a terrific tool to study interacting systems. Unfortunately, distribution analysis under non-ideal conditions becomes a very different beast. As you will recall, the C of S distribution was defined as a Fredholm integral equation of the first kind where we integrate over such sedimentation patterns of species with different S values. And we seek the combination of species that in their sum fits our observed data. In the case of non-ideal sedimentation, the sedimentation pattern of each species is described by a Lamm equation where the sedimentation and diffusion coefficients are not constant. Instead, they are concentration dependent, but in the case of a distribution, they will actually be dependent on all the species in the distribution and their mutual non-ideality coefficient as indicated here. And so, as you can see, in order to evaluate the sedimentation pattern of each single species, we would require already knowledge of the entire distribution. And this is a nonlinear problem. Due to the presence of hydrodynamic interactions, the species are no longer exhibiting independent signal contributions and the standard distribution analysis breaks down. Similar problems would be encountered with other methods, for example, dynamic light scattering. What can we do? There are two key steps to solve this dilemma. First, we make an approximation that the mutual non-ideality coefficients are all very similar. And this is not too different from the original C of S analysis that worked really well with a single average friction coefficient. And that would support that all species contribute to their backflow similarly. If Ks is the same, then we can pull it out of the integral and we are left with the integral over the entire distribution, which by definition is just the local total concentration. Second, we recognize that the sedimentation data themselves, if recorded with the interference optical systems, will give us directly an experimental estimate of that total concentration and its evolution in time. since conveniently the refractive index increments of different protein species are usually very similar. Now, under the hood, there will just be some numerical overhead to interpolate from the discrete experimental scan times to the time increments we need for numerically solving the Lamm equation. This is what we call a mean field approximation, where we exploit our estimate of the total concentration in space and time and an average non-ideality coefficient to predict the sedimentation pattern for each class of particles. This restores this into a linear problem, for which we can calculate the distribution. And once we have the distribution, we have the luxury of going back and replacing our initial estimate of the total concentration in space and time with a scaled combination of our sedimentation patterns. And in this way, the non-ideal C of S can solve the problem of coupled motion and breaks the nonlinearity of the distribution. And on top of the sedimentation coefficient distribution, we also get to adjust a best fit parameter for Ks and Kd. Let's look at an example. The circles are from interference sedimentation velocity data of 10 mg per mil of the NIST map, which is a very useful reference molecule. The solid lines are the best naive fit of C of S, not accounting for non-ideality. It arrives at a best fit frictional ratio of 3.5, which is of course way too high for an IgG, and caused by the fact that the standard C of S mistakes the boundary spread as a diffusion process and is unable to account for the self-sharpening of the boundaries. So not only is the boundary spread misinterpreted as a small diffusion coefficient, or correspondingly imposed a high friction ratio, but also the experimental data do not seem to adhere to the temporal spreading of a diffusion process and remain steep throughout. And this gives us a huge misfit of the data, with the correspondingly high RMSD of the fit. By contrast, the non-ideal C of S gives us an excellent fit. And not only that, for this analysis we fix the frictional ratio to what we would measure in dilute conditions and optimize the non-ideality coefficients. This provides us with estimates for Ks and Kd, from which we can calculate a second virial coefficient. We can corroborate the Ks value, which compares favorably to the value from an entire concentration series, and we found the virial coefficient is very nicely consistent with what we measure independently in a sedimentation equilibrium experiment. And here are the calculated distributions, in gray from the naive standard C of S and in magenta from the non-ideal C of S. In comparison, the non-ideal C of S provides us with a distribution of species as a function of their S value in units of S values at infinite dilution. It accounts for the existing non-ideality. By contrast, with the naive standard analysis, we would only measure the reduced observed S value. Applications in sedimentation velocity in the pharmaceutical industry are frequently aiming to measure small contributions from higher order structures. In the present case, we find that there is about 3% of a dimer in the sample. Importantly, unless we analyze the data with the appropriate model, taking into account the non-ideality and reach a good fit, we would miss that. Here is a different example. Apoferritin. Similar concentration at 13 mg per mil, but this is a bit more polydisperse. Again, we can get an excellent fit with our non-ideal C of S, and we obtain a Ks value that is within error identical to the result from an entire concentration series shown here, which was analyzed as an isotherm for the concentration dependence of the weight average S value. With the non-ideal C of S, we can get that number at once. In addition, this molecule does not show self-association, and therefore we can compare the distribution obtained with the standard C of S from dilute sample, which is in blue, with the result of the non-ideal C of S of the concentrated sample in magenta. This shows the polydispersity is nicely resolved. As a last example, let's look at a molecule that shows multimodal boundary structures. Here is interference optical sedimentation velocity data of BSA at a high concentration, 52 mg per mil. The solid lines are the best fit with a naive C of S distribution, not accounting for non-ideality, which gives us the last misfit as expected. By contrast, the non-ideal C of S fit is great. And here is the corresponding distribution in magenta. The blue and green curves are what we obtain at lower concentration. So this is very nicely consistent in the fraction of dimer and the ability to resolve even traces of trimer. As we discussed earlier, the Johnston-Oxen effect will mask the amplitudes of the faster sedimenting boundaries. This is illustrated here, showing as a dashed line the concentration dependence of the dimer boundary that is observed with a naive standard C of S. The Johnston-Oxen effect clearly is at play, suppressing the apparent dimer fraction. However, the filled symbols here are the results of the non-ideal C of S. And now the dimer fractions are essentially independent of concentration, and the same is true for the trimer fraction. This is a demonstration for how the non-ideal C of S will automatically account for Johnston-Oxen effects in the resulting distribution, because it is based on a detailed simulation of each species' sedimentation pattern. But really, this example at 50 mg per mil of BSA is a little bit pushing the limit of concentration where the non-ideal C of S model is really applicable. One problem is the optical detection in the analytical ultracentrifuge. When we create sedimentation boundaries with amplitudes of tens of mg per mil of protein, they will be associated with a significant refractive index gradient. So let's say we are trying to image this boundary. Light in the refractive index gradient is curving away, and therefore the position of the boundary image in our detector will be shifted. If we would be using absorbance detection with a slit in front of a photomultiplier, then the light could be entirely missing the slit. And that phenomenon has been termed black bands and is easily observed in steep boundaries of concentrated samples when imaged through absorbance optics. Yet another reason to use interference optics. However, here we still have to deal with the problem that the boundary position is shifted. Let me show you a movie of an interferogram during sedimentation velocity. We have loaded a lot of protein here that creates a pretty steep boundary that will be moving down the tube with time. And these gray lines here are shadows from little scratches on the top window, and they can serve as landmarks that stay in place during the experiment. Let me switch this on. What's noticeable is that the image of this scratch moves to the right just as the boundary comes through, and once it has passed, it is going back. Let me do this again. You see the gray band moving to the right of the yellow line and then back. What can we do? Well, the movie you've just seen already carries the seed for an experimental approach to deal with this. If you remember, the fringe profiles are always superimposed by a time-invariant baseline, which consists of adventitious blips and ripples stemming from imperfections of the optical elements on the nanometer scale. We can fit these constant signal profiles and usually forget about them. Compared to the signals from concentrated protein solutions, this TI noise is typically 1,000-fold smaller. However, the blips in the TI noise provide the natural landmarks that let us track the optical distortion. For example, here we have a blip that is initially in the solution plateau, which has no concentration gradient or refractive index gradient, and is correctly imaged. However, as the boundary transitions through, the blip will be radially displaced, and of course we have the added signal slope from the boundary itself. Once the boundary has passed, the image position will revert back to its original point. And here are some experimental data where in red is highlighted just such a blip. There is a Z-fit function that can fit the constant noise pattern, its offset slope, and radial displacement. And when we plot the radial displacement, we find it is in a good approximation proportional to the signal gradient. As a matter of fact, we found the proportionality constant, we call it a Wiener skew coefficient, depends only on the sample cell type, in particular the optical path length. It might also depend on the particular instrument, but this can be easily measured, perhaps when we calibrate our instruments. What do we do with this? You may remember that a key step for modeling hydrodynamic interactions was to evaluate the total concentration as a function of space and time for the mean field approximation. With that, it is straightforward to calculate the total concentration gradient as well, and then apply the same radial displacement to our fitting model to incorporate the expected distortions from Wiener skewing. You will ask, how much does this matter? Using our experimentally measured skew coefficient, here is a simulation how sedimentation boundaries of an IgG at 50 mg per mil in 3 mm optical path length cells will be distorted. In blue you can see the ideal boundary, whereas in magenta is the one accounting for Wiener skewing. Clearly this is a major impact and needs to be accounted for. If we carry out sedimentation velocity analysis at high concentrations, there is another major hurdle. You may remember that we started our discussion of nonideality with a consideration of the concentration-dependent sedimentation as a Taylor series, where we dropped all but the linear term to define a phenomenological coefficient Ks at the onset of nonideality. Some labs prefer to use a Taylor series in the friction coefficient, which leads to this hyperbolic form. It really doesn't matter because both will be the same for small concentrations, and at higher concentrations the underlying ad hoc truncations cease to be valid anyway. The actual concentration dependence has been a long-time topic in the sedimentation community in physics, and much work has been published extending the statistical fluid mechanics theory of Batchelor at the onset of concentration-dependent sedimentation to higher concentrations. Here are recent results from the SWAN lab at MIT, combining theory, experiment, and simulations to describe a concentration-dependent settling of hard spheres, which is shown here in magenta, and sticky spheres, which are in cyan. And here are the key equations. The expressions are framed as a relative velocity u as a function of occupied volume fraction phi. Sedimentation and diffusion are coupled through the structure factor. Stickiness is expressed using a backstat temperature tau, which directly relates the second virial coefficient to that of hard spheres. For small volume fractions, the expressions converge to Batchelor's predictions. Let's see how our linear approximations based on standard nonideality coefficients are doing. The black dotted line is the linear form, and the blue dashed line is the hyperbolic form. At low concentrations, they are essentially identical. But at volume fractions exceeding about 2 to 3%, they start to diverge and are poor approximations of the physics-based results in magenta. So at high concentrations, we should abandon the limited nonideality coefficients. It will be much better to use the concentration dependence predicted from statistical fluid mechanics. This can be easily plugged into the C of S framework with mean field hydrodynamic interactions discussed before. What do we do about the volume fraction? Therein lies a problem. We had previously noted that the relevant volume is that of a hydrodynamically equivalent sphere and therefore scales with the frictional ratio cubed. In this graph, we have given mg per mil concentrations in units here, which correspond to the volume fractions above, and this is for spherical particles. Clearly, if we have non-spherical particles, then the same volume fraction will be reached at lower concentrations, and this axis will shift substantially. And this is why we have much stronger nonideality empirically for more extended molecules. Because this is a rough approximation, let's say it's the product with a correction factor called reduced Ks, which we'll expect to be around 1. Why do we call this reduced Ks? Well, if we follow the definition of Ks as the concentration derivative of the reduced sedimentation coefficient, then we will find in the limit of low concentrations, our reduced Ks scales the ideally expected value of Ks we had previously introduced based on Batchelor's results. And in this way, we have incorporated the theory for hindered sedimentation, which should be applicable to very high volume fractions. Since we have abandoned the nonideality coefficients, we do not need to fit for them. Instead, we fit for the reduced Ks as an adjustable parameter, and optionally may fit for tau if we want to consider weak particle interactions previously captured in Kd. Combining Wiener skewing corrections and the hindered sedimentation models allows us to study sedimentation processes at very high concentrations. This approach is fairly new, but let's look at first examples. Here are data from IgG at 10 mg per mil and 46 mg per mil. It is interesting to note that the appearance of the boundaries is very different. As one would expect, they are steeper at higher concentrations and move slower. But also, the dimer boundary that's still clearly visible by eye at 10 mg per mil is very much compressed at 46 mg per mil due to the Johnston-Oxton effect. Nonetheless, the hindered sedimentation C of S can unravel these differences and arrive at a very consistent distribution. And here you see BSA at 140 mg per mil. This data was acquired in 3D printed 1 mm path length centerpieces. We can resolve the monomer from the dimer and find the dimer percentage is pretty stable. From the same fit, we learn that the reduced Ks is 0.66, which corresponds in the limit of low concentrations to a Ks value of 9.1 ml per gram, nicely consistent with the value of 8.4 ml per gram measured previously at lower concentrations. However, the slope of the concentration dependence decreases substantially to 3.7 ml per gram at 100 mg per mil, which highlights the nonlinearity of the hindered sedimentation and the importance to abandon the limited framework of conventional nonideality coefficients. Interestingly, the fit can be slightly improved further if we allow for slight stickiness of the proteins, corresponding to a weak attractive potential. This concludes the presentation of hydrodynamic nonideality. Let us recapitulate. Molecules take up volume and sediment in a viscous solvent and in a finite container. Therefore, backflow and hydrodynamic interactions are inevitable, and clearly the effects must scale with the number of sedimenting particles. Empirically, we find that for folded proteins, the effects become significant above about 1 mg per mil, although the threshold can be much lower for more extended macromolecules. What's happening is a retardation of sedimentation, boundary sharpening, and masking of polydispersity. On top of this, Wiener skewing from the associated refractive index gradients creates optical aberrations. So what can we do? We've noted previously that the direct modeling with discrete Lamm equation solutions, as appealing as it might sound theoretically, is an ill-conceived approach because of the susceptibility to experimental imperfections and over-parameterization. This will be exacerbated with additional non-ideality parameters. As an alternative, and this is the traditional approach, we can determine weight average sedimentation coefficients and study the isotherm of SW as a function of concentration. At the onset of non-ideality, we can do this simply by integrating the standard CFS distribution. The isotherm then can be modeled with additional terms for non-ideality. The downside is, like we have mentioned above for the analysis of interacting systems, that the weight average S value does not take advantage of the rich information contained in the boundary structure. A newer approach is offered by the non-ideal CFS, which explicitly models non-ideality and can allow us to determine a sedimentation coefficient distribution jointly with non-ideality coefficients. This can work well up to tens of mg per mil for folded proteins. It will reveal, for example, if at high concentrations new oligomeric species appear. If we want to study very weak interactions and proteins at very high concentrations, we need to take into account Wiener skewing and use a sedimentation model from statistical fluid mechanics embedded in the Hindered sedimentation CFS. This will give us sedimentation coefficient distributions and simultaneously a measure of the effective hydrodynamic volume of the particles and maybe their stickiness. And with that, believe it or not, we have finally made it through our list of topics of sedimentation velocity of interacting systems. Now, I hope this piqued your interest, and if you want to know more, please consult the references for more details and context or our book. If you are practicing any of these techniques, please consider joining our listserv in order to crowdsource for any questions you might have. Thanks for watching.